So despite everything else being a little bit less conventional and we're going to do hands-on stuff, the first 15 minutes is going to be kind of conventional, me saying a few things. So thank you all for attending and participating in this experiment. There's a pretty good faculty to student ratio here, so there's almost a faculty for every student. So again, feel free to take advantage of help and assistance here. My job is just a few minutes of introductory types of things. By day, I work in the University of Massachusetts Medical School in the psychiatry department, doing developmental disabilities, neuroimaging research, autism, developmental disabilities, and ADHD and things of that sort. By night, though, I work on many informatics projects, the first of which, that's a very long-standing project, NITRIC, the Neuroimaging Informatics Tools and Resources Collaboratory, nitric.org. Hopefully everyone already knows about that, or if you don't, you should become aware of that, since that's really a go-to place to find tools, to find data, to host tools and host data, and also execution and operating systems and computational environments. So that's a great general resource to help solve different problems. The other thing I work on by night is a project called ReproNIM, which is a Center for Reproducible Neuroimaging Computation. And so this course you're participating in today is part of the training and outreach effort of that initiative. And JB is head of the training core, and a lot of the staff you'll see are parts of that project, and they'll introduce themselves as they speak about different things. So those are kind of two of the big initiatives that are sort of behind this presentation today. So this is kind of preaching to the choir because you've all decided to be here. But give me a show of hands. Let's just confirm you're in the right place. Who thinks engaging in reproducible research is important? Yes, good. If you don't, then you might be in the wrong room. Or, oh no, you're in the right room because we have to convince you otherwise. More interesting question, by a show of hands, and I'm going to give you 5 options, so don't show your hands yet. If I found, just this year, all the neuroimaging papers that have been published, and PubMed suggests there's about 3,300 of them, how many of those do you think are reproducible? So on the one side, 90 to 100% of those you think are reproducibility. That'll be option A. B is between sort of something over 50%. C is something sort of between a quarter and a half, 25 to 50%. D is less than a quarter, less than 25%. And E, or wherever I'm up to, will be none. So how many think 90 to 100% of this year's literature is reproducible? Okay, how many think between sort of 50 and 90%, sort of on the high end of stuff? Okay, how many think sort of between 25 and 50%, sort of lower, middle end of the stuff? Okay, how many think less than 25%? Okay, and the very extreme less than 25% would be none. How many think none are? Okay, there's a couple of skeptics in the room, but that's the way it goes. So to some extent, I have my own opinion about that answer. We can't really talk about a precise answer to that question until we define a little bit more about what do we mean. What did I mean when I said reproducible? And we all want to do more reproducible research, but and if we want to assess if we are or aren't reproducible, or a finding is or isn't reproducible, we need to think a little bit about what we mean by the concept of reproducible. So there's a lot of different ways to think about reproducibility. And a lot of different people sort of define some of these definitions slightly differently. So I'm going to sort of set up an expectation for the types of terms I'm going to use related to reproducibility in this slide here. So reproducibility, in the way we present it, is really a spectrum. And in the context of a reproducible publication, we start from the concept of the original publication. That publication takes some data, performs some analysis, and creates some sort of result. The very primary element of reproducibility we'll refer to as re-execution. If you go back to a particular paper, can the same authors and the same data and that same method be done again? Now, anytime in this whole session that we seem like we're being mean or angry about the field, I have done everything bad that we're going to be talking about, so I've done that. And I'm sure I cannot go back to a paper from 2008 or so, and on something we did on the corpus callosum and ADHD or something. I do not know where that data is, really. I don't know exactly which version of the operating system I was using at that point in time. If you told me to re-execute that publication, I can't do it. And that's bad. It's not good science to be able to re-execute the things that we've written and put into the publication. So we start from this concept of re-execution. Do you know where that exact same data is? Do you know exactly what that analysis was? And can you generate a document that looks exactly like the thing that you published? That's re-execution. And to some extent, it's very difficult to think about reproducibility generally if we don't even have the foundation of re-execution. And so a lot of what you'll hear in the course is about re-execution and really setting up experiments in a way that supports that. At the next level, though, in the idea of reproducibility, really are types of generalizations. So it's interesting that we take a bunch of money and we collect a bunch of data, spend a lot of time, and we do one type of analysis and come up with one type of result and maybe write a paper or two. And really all that money and all that time and effort distills into just a couple lines, a couple conclusions, a couple claims, and maybe a short little table of some of the foci of activation or volumes of substructure. But there was lots of different ways that one could have achieved that result. So in this idea of generalization, we want to sort of take a scientific approach and really control our variables. We have data and we have analysis, so if we want to explore generalization, well, let's keep that data exactly the same. Let's now vary the analysis a little bit and see how sensitive our conclusion is to exactly the analysis that we did. So that way we're sort of scientifically probing the space of where our answer came from. Exact same data, let's look at how methods affect that. Similarly, you can flip that the other way, take the exact same analysis and change the data a little bit, add some new data, use someone else's similar data. How stable is the answer, is the conclusion, to changing just one thing, just changing the data that's going into that analysis. So this gives us really, from any one publication, this gives us a whole sort of generalization space where we can systematically probe the sensitivity and stability of that answer as we, as scientists, twist our knobs of how we do the analysis or how we put in the data. Because ultimately, what we want in terms of reproducibility is that a nominally similar set of data, so instead of my kids with autism, it's your kids with autism, with a nominally similar analysis, not free surfer, corpus callosums, but FSL's, corpus callosums, should yield a similar result. The volume of the corpus callosum is smaller in kids with autism. The problem in that statement I just said, of course, is the word similar. How similar does an answer have to be, to be reproducible? And if I wanted to make my paper look novel, and I want to talk about, say, the default mode, well, default mode's kind of a wishy-washy set of regions, and I can either make my result look more similar or less similar to prior literature, depending on exactly how I want to define default mode systems or things. So again, left to the whims and wills of the authors, exactly how similar or dissimilar things are open to interpretations, and so that's something we need to be a little bit careful about. So I'm going to restate one of the premise here, that a true finding should generalize, i.e. always be true. If we reach a conclusion, such as the volume of the corpus callosum is reduced in children with autism, that statement means any valid way of looking at the corpus callosum should generate a similar decrease in the way I stated that claim in all children with autism, if it's really about autism. Now, in general, most of our publications do not really support that kind of conclusion. We don't really know exactly to what extent the tool matters. We can't really mean with my 100 or 1,000 subjects even that it generalizes to all of autism, a very broad disorder. And particularly these more spectrumy types of diagnoses, the subjects with autism who happen to be able to sit through an MRI scan may not be completely general for all of the autism spectrum, for example. So anyway, so we need to not stop publishing things. We just need to be more precise and complete and make the entire process of publishing things more transparent. We're a little bit late start. Reproducibility has been a problem. It's a problem not just for neuroimaging. It's not just us. It's not just me, being bad in the past. A lot of the basic science findings that lead to drug companies to develop try to develop new drugs, come from the basic science researches, and many of those fail in their development And so one of the drug companies, in this case Bayer Healthcare, tried to go back and look at all of the sort of basic science that was going into a number of their publications, a number of their explorations, and they discovered that a huge fraction of the basic science that are driving these discovery systems were not themselves re-executable or reproducible. So not only is it hard to get a good lead turned into a drug, it's infinitely harder when half of your leads are not reproducible themselves. And particularly in the domains of mental health, Tom Insel, the past director of the National Institutes of Mental Health in the United States, has been really hard on the field in that today a child with autism or an adult with schizophrenia who walks into their clinician's office pretty much has the same outcome and the same interventions that we had 30 years ago, before billions of dollars of research and in genetics and imaging and all that kind of stuff has happened. The outcomes and therapies are very similar to date. Now, a lot of scientists have received a lot of grants. A lot of scientists have written lots of papers, but the outcome for a child with autism or an adult with schizophrenia or a teenager with a drug addiction is really not been radically moved by this huge investment. There's lots of reasons for it, many of which are biological, and many of which are the subtleties of biology. But part of it's the technology and the information that we're using as part of the research efforts. Okay. When we generally think about a neuroimaging experiment, we have at least two streams of input data, one of which is the imaging data we get from our scanners and our subjects. The other is probably some behavioral and clinical assessment types of things. We bring those two sources of information together, we do a bunch of processing, come up with some answer, and we publish that. But when the final output is just the PDF of the article that you've just written, there is a lot of wiggle room and variance in terms of exactly how did that data from the subject, clinical and biosamples and things, turn into the data you're using, exactly how did the data from the MRI scanner turn into the data that we're working with, exactly what process you did, and exactly what pieces of those results you chose to publish has lots of variation and are not completely described by what we put in our publications. This just reminds us that this sort of workflow has lots of different elements to it, from the data to the publication. And what we've learned over the past few years is that everything as part of the experimental workflow matters. Exactly what operating system you're using matters. Exactly what tool you're using matters. And exactly what data you're using in your experiment matters. All these things matter. And so all of these things are what we as experimentalists now need to take a little better control over. The other sort of guideline to what we'll see from the other presentations is really a vision towards this sort of next generation of what a publication should look like. The era of just the words, just the PDF being the output of your research effort is really hopefully coming to an end. You and your students will hopefully really see a new era emerging of what a publication and what the product of research begins to look like. So in addition to the lovely words, which we need in order for humans to understand and comprehend what's going on, we need to encapsulate the actual data that went into a publication, the actual workflow specification that goes into a publication, the computer system, the execution environment, as well as the complete set of results. Now, the opportunity here from the research and the research efforts point of view is that what used to be one publication, one entry in your CV that I did this study of neuroimaging, can actually turn into more like 4 publications, 4 research products of your effort. There will be the lovely publication that you write about the conclusion, but there should also be a publication for the data. There could be a publication for the workflow you're using. There could be a publication for the results that you're making available. And all of these things are very valuable as the product of your research enterprise to make available and to get the scientific credit for. We've got to move scientific credit from just that publication to the scientific credit for generating this information, getting that information into the analysis ecosystem and reused and built upon. So that's good news, ultimately, that we should be able to document and get more credit for the different elements of the scientific process that we're engaged in. This course, as JB sort of foreshadowed, is just a day-long sort of workshop hands-on of a much larger training initiative. The ReproNim Training Initiative, you can find at training.repronim.org. The 4 main modules that that are in that course will be presented today. And this course is designed really to follow the software and data carpentry modes of really ultimately training the trainers. We cannot go out and train everybody in all this kind of stuff. We got to train you guys, and you guys then got to train the other people in your labs and really become part of how this movement keeps moving. The other thing to point out here is that full curriculum, each one of these modules can take a month or 2 months to do. It's not just a little bit, and a lot of information is being distilled there. A lot of mechanisms are being purported to you. So when we attempt to do what's a sort of month-long course in an hour or an hour or 2 for one of these sections, we're only giving you the tip of the iceberg. We're really only trying to motivate you to want to go and do the rest of that course. So to some extent, some of the comments we'll hear about this is it's going to be, hopefully appropriately, overwhelming. Because to some extent, we want to remind you that you can't just learn reproducibility in an hour or a day. We want you to be aware of the issues, but also motivated to go and continue the training in terms of understanding what you need to know to improve your reproducibility of your research. And again, the other balance is to do everything right can be very overwhelming, and one might want to just throw up their hands and say, I can't do that. I'm just going to go back and do things the way I used to. So the other thing that I want to reinstill is that one of our goals is to find as many simple things that we can do that make our reproducibility better today than it was last year. We don't necessarily have to get to the very end right away, but to the extent we can find simple things that we want to do and find simple things that we can do that moves us along this, the whole field overall will get better. So that's think reproducibility and act re-executably. So you'll see 4 different modules presented. Again, JB foreshadowed what those will be, and you have it in the documentation somewhere. And so I think, without further ado, we're just trying to make sure that we all have the more and more tools to keep from going off the scientific tracks in terms of our overall research efforts. So without further ado, I will turn things over to the first presentation. That's Jeff Gretta from UCSD, University of California at San Diego. And while we change computers here, there will be an opportunity to make sure everyone's computational systems are happy.